Okay, good afternoon everybody, and it's my pleasure to tell you how Microbiotica is translating precision in the microbiome science into transformative medicines and biomarkers. And on this slide I'm going to summarize the story. We firmly believe in Microbiotica that the microbiome needs to be studied in human beings, and that means patient groups. And we undoubtedly have the best-in-class patient-first microbiome discovery platform. And that's because it comes out of 10 years of investment at the Sanger Institute. This makes us the global leader in gut bacterial culturing, reference genomes, and microbiome AI technology. It gives us an unrivaled microbiome analysis linked to patient phenotype for the discovery of therapeutics, biomarkers, and targets. And on top of that, we've built leading translational capabilities and CMC preclinical development so that we can take products through to the clinic. Our business model exploits the platform in three areas: live bacterial therapeutics, biomarkers, and target discovery. And for our internal pipeline, these are always supported by major academic clinical collaborations which bring in large, high-quality data sets to feed the analysis. Our current focus is on immuno-oncology and IBD, but we're entering new areas. And within 18 months of forming the company, we had validation from a large deal with Genentech, which at the time was the largest in the microbiome area. And this is a major global clinical study in IBD. We're growing a pipeline of best-in-class differentiated products, multiple therapeutics and biomarkers. 2 will be in the clinic in 2021 in phase 1B. And our immuno-oncology and ulcerative colitis programs are based on high-quality clinical data sets and unique bacterial signatures that others cannot find that we achieve from our platform. Now let me just give you a little bit of introduction on the microbiome, and this is primarily the gut microbiome. The bacteria in our gut, more than 2 kilos of them, they influence every organ in our body. And while a few years ago the links were only made to a few diseases in the gut, there are now firm mechanistic links to diseases and conditions from brain to cancer. They're also importantly a key determinant of drug response, very importantly in the checkpoint inhibitor immuno-oncology arena, where there's tremendous excitement. With firm evidence of causality, mechanistic links, and data coming through from companies that are at the vanguard of the modality of live bacteria makes them amenable to therapeutic intervention with sustained effects. So this is a major emerging multi-therapeutic opportunity, a new modality, if you like, for the company that can make sense of the science. But therein lies the but. Our strategy and vision is to be the world's leading company in understanding and stratifying the microbiome for the discovery of therapeutic strategies with 2 sides to the coin. We work by finding bacterial signatures linked to phenotype, which give us the bacteria that constitute live bacterial therapeutics, best in class, from high precision patient-first discovery. And the other side of the coin is that those bacteria are both a biomarker and you can walk from those into targets for conventional discovery. Now, we're not just making this up. We're underpinned by over 10 years of investment at the Sanger Institute, which invested in Trevor Lawley when he was brought over from Stanford to start the microbiome effort, and was funded for this period to address some of the fundamental barriers to this field. The barriers included rational design of live bacterial therapeutics to replace fecal transplant. There's a paper at the top. There's the mass culturing and isolation of gut bacteria, the analysis of gut bacteria by metagenomic sequencing, and the precision you can achieve when you have reference genomes. And that was applied most recently in a major birth cohort study, the first example of this mass analysis, which we're undertaking with Genentech. And Genentech had been watching this field for 4 years and have documented in script that they were looking for the fundamental rigor to enter the microbiome arena in IBD when they did the deal with us. Now let me tell you what we actually do, and this outlines it. It's based on our unrivaled patient stratification capability. We start with large patient groups, data sets and samples, sufficient to give us the statistical powering to draw conclusions when the bacteria differ in different individuals. We apply unrivaled characterization of the bacteria and unrivaled precision to strain level characterization. We then apply microbiome machine learning tools with all the metadata and outcome data, and from that we get bacterial signatures of consortia of bacteria acting together linked to phenotype. If it's checkpoint inhibitor drug response, then those are a predictor of response to, say, Keytruda, or they are the bacteria that you would give to turn a non-responder to Keytruda into a responder. Here. Now, I'm just going to have a few slides on the platform before telling you about the pipeline. It's all fundamentally based on our ability to culture bacteria and generate the world's largest reference genome database. This is, if you like, the landscape of gut bacterial genomes in the same way that there was the Human Genome Project. Microbiotica has the biggest portion of this real estate, 5 times the rest of the world, and that allows us to do the precision from shotgun sequencing. You have references to map the sequences against and identify the bacteria. It's linked to a culture collection, a very closely linked to a culture collection. Another key technique, which again is unprecedented, is the ability to culture all the gut bacteria in a few weeks from any one human. We call this personalized bacterial bank, and it's characterization by isolation. We've done it about 200 times, and it gives us a complete gold standard fingerprint for each person. Importantly, gives us those bacteria ready to progress as therapeutics. So we apply this, for example, to exceptional responders in checkpoint inhibitor clinical studies. This slide shows that whereas we can assign 95% of sequence reads in shotgun sequencing from patients, with public references you can assign 60%, and a leading commercial provider assigns 50%. That means the rest of the world is assigning 50 to 60% of the bacteria, and we're seeing all of them. I think I'll leave that there. The other one actually is Trevor Lawley's gut bacteria, and showing the number of species and strains we get. We can see twice as much that everyone else can, to completeness and with precision. And this shows a technique that allows us to identify bacterial signatures by machine learning. It comes out of a fecal transplant study which causes remission in ulcerative colitis, and we're finding the bacteria that do that. And here's some individual bacteria using the techniques I mentioned. We find that in blue, each of these circles is one bacterium. In blue are the non-responders. In yellow are those who go into remission or cure long-term. There are differences in abundance for those bacteria, but when you apply our machine learning tool that combines all the different combinations of bacteria in different numbers, we find a combination of 10 bacteria that is more or less 100% predictive of remission. And so that is a perfect biomarker, but also a candidate drug, because those bacteria have gone into everyone that's responded and been cured, but not the others. This is our pipeline. I've touched upon ulcerative colitis. That is a collaboration with Adelaide with an option to Genentech. We have a big activity in immuno-oncology, an asset in C. difficile. We're starting 2 new programs, and we have our big biomarker discovery programs, one with Genentech and one with in cancer. Now, the Genentech collaboration, this is the world's biggest microbiome clinical study. It's looking at 2 development assets in IBD in 4 clinical studies over 40 countries, and we're characterizing and identifying predictive signatures. With that, we're increasing our collection of gut bacteria and adding to our platform. Now, I just want to very briefly finish by touching on immuno-oncology. This area is the biggest growth area in the pharma industry, led by Keytruda and Opdivo. Sales will be reaching over $55 billion from nothing in just a few years. They're transformational and they work for many cancers. More and more are being registered for more cancers, but the patient response rates are still terrible. 40% is the highest in melanoma. And so there's a major opportunity for co-therapy to increase the number of responders. The remarkable thing is the gut bacteria control the response to checkpoint inhibitor drugs because they control the immune system tone. Antibiotics catastrophically reduce the response rate. There is mechanistic data showing that the whole microbiome from patients that respond transfers response in mice. But the groups in the academic sector do not agree on which bacteria are involved. And we're developing a best-in-class product based on the leading microbiome clinical data and our microbiome profiling technology. Will be in the clinic in a phase 1B in 2021 with a collaboration to follow with CRUK and Cambridge hospitals. This slide summarizes it. 3 leading groups that have been publishing in Science and Nature since 2015 came up with completely different bacteria when they tried to dissect which bacteria confer response to checkpoint inhibitors. These could not be more different. The microbiome profiling is not strong and these studies are not well powered. The data has been released into the public domain and we and others don't find them to be statistically significant in showing a difference between responders and non-responders. And competitors in this field are rushing to the clinic with bacteria as co-therapies based on different hypotheses from these studies and in vivo studies in animals. But based on our MELRESIST study, which we have the commercial rights to, we've identified an unprecedented signature which is 90% predictive of response in melanoma, not just in our study, but across all 4 studies. So we found what's missing in the sector. And from that, we've got a signature of 9 bacteria, which are the lead that we're taking forward that correlate in all these studies. This is the team. Trevor Lawley remains as a faculty member at the Sanger while being CSO. He's undoubtedly a leader in the microbiome. This is my second company. I've also held major roles in GSK. We've recently appointed a very experienced chairman with experience of the US public markets because that's where we're heading. And we have a team of 40 with experience from both competitors of the microbiome and big pharma. And I'll leave it there.